Thanks everyone for being here on Valentine's Day. I hope you love the presentation. So I decided I'd start out if people are here by mistake. I want to make sure I give them a chance to escape. When Chris originally asked me to talk, and thank you for the invitation, Chris and CAIRIBU, she had suggested the idea that we talk about budget, and I can't think of anything more painful than talking about preparing budgets for an hour. So I broadened it, and I said, I think it's important that we talk about lab management. And I really was thinking about this from the perspective of when I started a lab, what I wish people would have told me. So I think this is probably most relevant for newer emerging independent researchers, but maybe also for trainees that are looking for ways to improve their organization, or for experienced investigators that are looking for some tweaks, or maybe just to start over altogether. So when I think about this, I think you really only get one first chance to do it right. And that's the way I entered when I was starting a new lab. I see a lot of new faculty members sprint to get the things, all the equipment, all of the supplies, to fill the shelves, to make a lab look like a lab. And then they start into their work. But for me, what I thought was most important, and I agree with that now that I've gone through the process, is to think about the process itself and spend a lot of time into thinking about how things will work, not just which things you're going to occupy in the space. And it takes a lot of time to do this, to really think through every aspect of what you plan to do in your ideal lab. It's really a marathon. It's not a sprint. So this is going to be a guide for you to develop your marathon plan. I am not a runner. I think the first thing that you want to think about, and this is kind of with all aspects of research, of teaching, is to really think about what that finish line looks like for you. I started in a tenure track faculty position, and most important to me was to be successful in getting tenure. And so you might not think about this, but it's really a great idea to start with that end line, find out what the guidelines are for that end line. These are some of the guidelines that were listed for the UW-Madison promotions process, and what you would have to actually submit and prepare when you're thinking about lab management. So I had a good understanding from these guidelines and from talking to people about expectations of the size of the lab that I might need. I knew that it was very important for me to train graduate students and undergraduates and postdocs, as opposed to having a laboratory consisting solely of scientists, because that mentoring is part of how I would be evaluated for promotion. So that told me a little bit about the kinds of people that I might have in the lab. And it also told me a little bit about the size of the lab that I might need. I had a good understanding of the requirements in terms of number of papers that I'd have to publish as senior or corresponding author, the types of grants or how many grants I might need to be successfully awarded. And that shaped the way that I thought about our lab, and especially the process that would go into developing, building, and sustaining a productive environment. These guidelines, whatever it is that your finish line looks like, is unique to everyone. So spend the time to look at wherever you are, what success might look like for you in the end, and then build from the very beginning towards that ending goal. Of course, with any competition, there are rules, and there are absolutely rules for building and maintaining a lab. We're familiar with a lot of them. There are institutional, state, and federal rules that guide how we do research, and they're really unique to the types of research. I can't believe how many protocols I had to fill out and continue to have to fill out new in terms of covering all of the types of things that we do in our lab. We use controlled substances. We've used a select agent. We use rodents for research. We have a human subjects IRB protocol. We work with hazardous chemicals and had to work on a chemical safety plan. All of those things take a tremendous amount of time. But if you know what the institutional rules are, you can build your management plan accordingly. And it's not just those types of things. There are other things that we don't think about. I flashed a couple of those here, but there are spending rules for your institutions. For UW-Madison, for example, we have contract vendors that we're supposed to work with. And if we have others that are listed as do not purchase vendors because they didn't meet compliance for UW-Madison. You need to know that before you start ordering supplies. We have purchasing rules. Limits on how much we can purchase with a single purchase, spending limits on our purchasing credit cards. It's useful to know those things, not only for you to start by yourself, but if you intend to hand some of this work off, and I highly suggest that because it's too much work for one person, you need to know and be able to build guidelines to share appropriate spending and purchasing procedures with others in your lab. So that all goes into lab management. Knowing these rules are really important. Think a little bit about what your perfect laboratory might look like. It's different for everyone. We all work on different things. Some envision a huge lab with many people working hard. Some want to work in a smaller, intimate setting where they work with a small team and get to know that team very well. Others, maybe you're going to be working on your own and it's data processing or something like that. So really think about it. This is actually a picture of a new lab that I plan to move into. It was supposed to happen this summer, but it's been delayed and delayed and delayed and delayed. So someday, maybe we'll have things on the wall and in the drawers and people working in this space. One of the things that we don't think about, again, we're hurried to get the reagents in and start the experiments, but take a moment to really think about how you want to structure your lab. And this really, I think, is individualistic. Some people like to be involved in every aspect, a true top-down type organizational scheme. Others want to be peripherally involved. Others want sort of a middle person who can organize and maintain the lab for you. And that decision isn't just based on your own personality, but it's also on the type of position you have. Are you going to be someone with high administrative duties, or are you a practitioner that your time in lab will be limited? You might not want to be the one that everyone comes to every day with problems. You might want a middle person in between there to help offload some of the burden, but also to give consistency to the people within the lab. Another thing that you might want to think about when you start a lab is how your resources are going to be maintained. Are they going to be maintained by individuals, kind of a siloed approach? Or is it going to be one big supermarket where everyone can collect and maintain and restock as needed? Now, there are trade-offs for this. With the siloed approach, there is research duplication and cost. It puts the responsibility for success on individuals or within projects and not on the entire group. With the grocery store approach, where everything is shared, there's some real advantages. Everyone can learn how to operationalize a lab. That's an important skill, especially in somebody in a training position. People in my lab participate in purchasing, they participate in budgeting, maintaining the budget, restocking, preparing buffers, but there's a potential cost or drawback with that type of model in that one bad egg that doesn't restock, that doesn't do their dishes, that doesn't reorder, or think to even note that something's been used up, or uses everything but 1 microliter. One bad egg can spoil the entire salad. So you have to think about what you like and then how you're going to organize your lab and maintain things based on that structure. So in terms of my own preference, I didn't want to be in a hierarchical structure or a flat structure. And I was concerned that it would be too time consuming. Every single problem comes to me every hour of every day. Doesn't give me any time to do my writing, teaching, other administrative roles that I have on campus. And so I prefer what I call a modified network structure. And that's showing over on the left-hand side here. I think of myself as the gray circle in the middle. And then there are 3 project teams surrounding me. I like the idea that individuals each get a chance to lead a project in the lab. Sometimes, depending on the size of the project, even some of our younger trainees, undergraduates, for example, it's important to learn leadership skills, but it's also important to learn how to play as a member of a team. And so I like to structure this in a way that individuals participate on multiple teams, sometimes as a leader, sometimes as a team. And I think that that's consistent with the way most of us experience life. We're not always leading something. And so you have to develop both types of skills. And I think you can see frustrations that emerge as a leader when your team members aren't doing what you want to do. And maybe that makes you a better team member and vice versa. So to maintain this structure where I have this modified network structure, everyone gets to take a turn at leading, and to maintain a lab where we have the supermarket approach and everyone shares resources, there's some things that have to happen to maintain effectiveness of that type of environment. First, there needs to be clear expectation for leaders as well as team members, and how those individuals interact to create their products. This modified network structure does create certain challenges when it comes to ownership of publications at the end, for example. The team leader might serve as the lead author or one of the lead authors, and the other team members are co-authors. But if you look at this in a siloed approach, each individual might get one or two publications, but in this modified network approach, everyone can participate and be listed together. So I think getting buy-in is something that's important. And the advantage of this structure is that you can be more productive. It's important that we work from interchangeable and interoperable protocols. When we're working across teams like this, we want things to be seamless. If every individual works off their own unique protocol, we can't mix and match data. And this has happened to me when we come to the time of publication, where we start to write and realize that somebody did one thing and somebody did it another way. We have to redo the experiment to make it consistent, and that's never efficient. So that's a really important thing. Having findable data. Members of my team work on different schedules. We've had high school students who can only come in after school or on weekends. We've had undergraduates who are occupied with classes most of the day and sometimes prefer to work at night or early in the morning. We have others that are here all day long. They're not necessarily here at the same time. So having a schema where we have data that's findable for others to see is also very important for us. And I think everyone needs to have a shared vision of what that final product would look like. If we're not working towards a common goal, these networks fail. So I mentioned I use this modified network approach in terms of our organizational structure, but I've found that over the course of the time that I've been in lab, now it's approaching 15 years, that I've led my own lab, but much longer working in labs. The course that you're plotting, it has some unexpected things that are gonna pop up in the future, and you need to be prepared to adapt. And so the structure that works for you today, or when you start a lab, or now that you're maintaining a lab, isn't always what you're going to see tomorrow. Labs grow in size, they shrink. Sometimes it's dominated by junior individuals who need a lot of extra guidance. Sometimes there's senior individuals who need space, and sometimes it's a mix where it provides opportunities that you can change the structure so that your senior people are teaching junior people. So adapt. And I've seen that myself, and some of the things that I set up initially stopped working, and we had to think differently. So what I'm showing you today is not something that works under all circumstances, but it will give you some ideas in terms of what you can do. Okay, the next step in our overall marathon is to set interim milestones. It's so exciting to sit down on the first day that you open your new laboratory and you can call it your own. I remember talking to a really cherished colleague and mentor of mine before I started my lab, and he reminded me of when he started his lab, and at that point it had been like 40 years ago. And he said, I walked into the middle of the room, I opened every drawer and every cabinet, I sat down on a chair in the very middle, and I thought to myself, what have I done? He thought about all the things that needed to start that were just laid out for him as he had been doing a postdoc, and now would need to be redone. And it's daunting. It's daunting to know where to start. I think there are two types of personalities: those that dive into it and try to get everything done as fast as they can, and those that sit back and plan a little bit. Maybe three personalities, because I've seen some that sit back and plan for so long that nothing really gets started. So I think a good thing to do is to set some interim milestones. You know what your finish line is going to look like. You've already reviewed that and thought about it. Now, what are the things that you're going to do when you get to mile 5, mile 10, mile 15, mile 17? Mile 17 hits when your startup funding starts to go down and you start to get GI issues and can't sleep at night. Have a plan and then execute that plan. So when I started, it was a little bit of a trick. I remember thinking, I'm going to get ahead. I actually did a postdoc at UW-Madison, and I thought, since I know this institution, I'm going to be the fastest person to ever start as a faculty position, and my publications will come flying out. It wasn't exactly true. I thought I knew what I needed to do. I have to write an animal protocol. I have to write a biosafety protocol. I have to write a chemical hygiene plan. But I opened those protocols up, and I realized the first thing it asked for was my room number. I had that. And then it asked for a telephone number. And I thought, I don't have a telephone number yet. I'm not sure who I talked to about getting a telephone number. So 10 phone calls later, I found out the person I had to talk to, and I called them, and they said, sure, we can get you a telephone number. Do you have a telephone? I said, no. They said, okay, well, you need a telephone first before you can get you a telephone. So another 10 phone calls. How do I buy a telephone? Well, we don't have your account set up yet. Wait a little while. It was a couple days later, the accounts are set up. Okay, now you can buy a telephone. Well, what do I use? Oh, well, you need to have a purchasing card. I need a purchasing card. How do I get a purchasing card? So we went through that process, another couple of days. Oh, to get a purchasing card, you have to go through 5 hours of training. So I went through the 5 hours of training. Finally was able to buy a telephone. Got my telephone, and I went into the lab, and I'm ready to fill things out, and asked me where the fume hood was and when it had last been certified, and whole thing started over again. So one of the things to think about in terms of your schema and your management is how you get started and how you set these milestones. What is the order of operations? It was so frustrating to me that it hadn't actually been relayed, and I thought about all of the people who had gone through this process before, within my department or even within the school, that I sat down that day and I thought, I'm going to write a timeline, and I'm going to pass this back to them so that anyone who follows me as a new assistant professor doesn't have to deal with these frustrations. So I did that. And it turned out that that protocol, again, part of management, was so important to the vet school that they said, we love this. We are going to publicize it, and we're going to take it a step further. We want you to be part of this group that tells us what somebody needs from the very beginning and how we can best get them to the finish line. So I sat down and I was in part of this mentoring task force, and we went through the entire School of Veterinary Medicine. I don't know how many people, faculties, at assistant, associate, and full professor levels, and interviewed them in groups and learned what it means to best onboard somebody and usher them through the process of going through their promotion and obtaining tenure, that we were able to write an entire document. The document contains a checklist of what to do in order, but it also says, here are the things that you should be thinking about in terms of your professional development and the milestones that you should achieve at each stage. Again, important in our marathon to have these milestones written out, and they affected how we did lab management. This is just an example of some of the orders of operations with the phones here that we did. So I have a series of documents all prepared in a file, and I'm working with Dr. Peniston to get these uploaded onto the CAIRIBU website so that people can use these as templates. Obviously, at your institution, things are going to be different than they are here, but they give you some things to think about in order of operations. Okay, so a big part of having our modified network schema work is to have clear expectations for everyone. So I was fortunate to be urged into forming a expectations document or a contract. This was long before I had taken all the mentoring classes that told me that this was the best practice, but it was from a graduate student that was working in my lab that was taking a course on mentoring, and they had suggested that this was a really good idea. And so I sat down and with that individual made an expectations document. It lists expectations that I have of anyone who's working in the lab, and it doesn't really matter what level they come in at, whether it's undergraduate or postdoc or scientists. I also have a series of expectations that they can expect from me, what I promise to deliver to them. And this is something that I hand out to individuals who are doing graduate students who are doing rotations in the lab, or others who are considering joining, and then I have them read it and I say, you know, this is what our environment's like. I just want you to be aware of it, and this needs to be something that you buy into. We give an opportunity for individuals to add things or discuss things that are confusing. So this puts us all on the same page, hopefully, to build a great and functional network. I mentioned that it's critical that we all work from the same protocols. And this has been successful for me, and it was born out of a frustration I had as a graduate student and later as a postdoc, when I came into a lab and I wanted to conduct my research in the way that they conducted their research so that we would be unified. We could compare historical findings to ones that we have today. We have an understanding of whether things are working or not, and we can compare and contrast our different experimental variables. I struggled because those labs that I had been part of, everyone did things their own way. Some of them were written on the back of a napkin. Some of them were memorized by somebody's head, and they would show you once and you would be expected to know it. And that resulted in problems because everyone was working slightly in a different way, meaning that we couldn't share each other's data very well. And so we standardized how we made protocols. I came to realize after some time that this is something that actually can help to build your reputation as a researcher and as a laboratory. And so we spent time branding. We came up with a little lab insignia that we would use and stamp onto the top of our protocols. And we would share them widely. Sharing research instead of keeping it inside was really helpful to my success as I went through the years, because other people got to learn that things worked, maybe learn that they worked well, and that helped to grow our reputation, and it built collaborators and eventual co-authored grant applications that were successfully funded, and publications, and even students that we've shared across collaborating labs. And so I highly recommend you open yourself up to the public. I think when we specialize in something, we have a lot to offer others who aren't necessarily using that technique as their primary thing, and it's great ways to grow. So we modeled our protocol after Nature Protocols format. We've used this to publish protocols on various sharing websites, and we hand these out freely. We also, as part of the expectations document, ask that everyone in the lab follows this protocol. And if a problem is identified, or if an improvement can be made, we make that improvement, we change the version number, and then everyone follows that new version. So we have a way for adapting as we go through things, but this ensures that whatever Dr. X does, Dr. Y is doing it the same way. And if there's a chance that something that they've done previously or into the future can work together to form a manuscript, their productivity is enhanced by that. So another part of having this network function successfully is that everyone is able to find data and interpret data. I remember we spent weeks trying to figure out how we would name files so that they were predictable and that everyone would be able to find them. Again, out of problems that we've had when early on, I'd see students would come in, would work independently, would save their work, and then when we go back in to try and find out what they did later, it was so confusing how they named their files and where they put things that we couldn't find half the things that we knew were there, even looking through lab notebooks. And so thinking about how you will standardize electronic data and make the name meaningful can really save a ton of time later. These are things that you need to go out and pull for a grant application, for example, or for a publication. You want this to be understandable to you as the lead of the lab. We also spend a lot of time thinking about how we organize data. We have available to us a network drive that gets backed up daily. And it's important that we keep that data secure. So it's a secured site. Everyone is able to use their own independent folders on that site. But when it comes time for publications, we have them duplicate their data and drop the data in a folder so that each publication we've ever submitted contains all of the data and metadata associated with that publication. Makes it really easy if a reviewer comes back and asks for something that you can show them a different image or a different data point. And again, the one additional advantage is that when you template out figures, whether it's a 6-panel figure or 8-panel figure or whatever, a new person coming into the lab can use that same template and there's consistency. Where I find that that's very useful is later on when I have to give presentations, everything's using the same font, the same size, makes things look a little nicer. Even with a consistent naming architecture and files placed in certain locations, it's still sometimes difficult to interpret what you're actually looking at. And we do a lot of multi-fluorescent immunostaining images, and it's not always clear to me what antibody was used to create the green channel, what antibody was used to create the blue channel or the red channel. So another thing that a scientist in the lab had started to do and was a great practice was to drop a text file into each folder. And that text file contained the metadata. This is kind of what it would look like. This is an example that came out of one of them. The advantage of a text file is that as operating systems are updated, the text files are something that can be read consistently through time. And so it's an indelible piece of metadata, and it helps to explain what was there, maybe where you would go in a lab notebook to find out more about the experimental design. Very, very useful. And so dropping these into data folders can accelerate productivity. We spend a lot of time thinking about how we store our archival samples of a lot of different varieties. We spend a lot of time doing histology, and so we have hundreds of boxes of microscope slides, and without much information, it would be impossible to find a sample that we needed. And we need these all the time. Maybe we're needing a specific tissue to use as a positive control to test a new antibody. Or maybe there was some experimental design involving a mutant animal, for example, that we can go in and mine those tissue samples to test new hypotheses, but only if we can find them. So we created a scheme where individual tissue blocks that were used to create the sections were given a unique identifier. It's a 4-digit code shown here on the left-hand side or the right-hand side. Who was responsible for collecting those samples? If they were cut into tissue sections and stored on a microscope slide, which box number they were located in. And then we have information about experimental design. And we like to do multiple samples in the same block so that when we have microscope slides, one row might be our control samples and another one might be experimental variable 1, or another one might be experimental variable 2. And by arranging those in specific types of arrangements, you can make them so that they're asymmetrical in all directions. So that if the tissue section gets flipped over, it doesn't matter. You'll still be able to decode and know which one is the control and which ones are the experimental variants. And super helpful. We've been able to go back and generate new figures from tissue sections that have been sitting for 5 or 10 years just because we collected and organized the data so efficiently. We do the same thing for samples that aren't necessarily stored at room temperature, but that we store in the refrigerator or freezer. And so we've made these little schematics of what the inside of refrigerators and freezers would look like. We give names to the location, numbers and letters, combinations. And then we have a spreadsheet where we list what specimen is located in each position. We're collecting information like who collected the sample, maybe where you can find out more about the experiment, a page on a laboratory notebook, the age, sex, species, strain, tissue, orientation, other treatments that might have been used. And lab mentors can use the search and find function in Excel to find relevant samples or to know that we don't have them. This saves so much time and so much money. And again, increases our publication number and our productivity. Makes it really easy to collaborate too. I've had people that have said, hey, I saw something in one of your publications. You wouldn't happen to have that anymore. And I can find it right away and hand it off. Might result in a co-authorship for somebody. Especially if they added their name to it, we can get the students involved in that as well. I like to be creative and create art, digital art. So these are some of the things that we've created to either be part of our protocols or to be part of presentations or to go into publications. We have a lot of these things, and students generate a lot of these things as well. One of the problems that exists now is that a lot of those individuals keep that material to themselves. So everyone needs to reinvent the wheel. We have a community data repository where if you invent something, some digital media, you drop it in there and you name it, and then anyone can use it. And we also have a repository where we put PowerPoint presentations. Again, anyone can go into those PowerPoint presentations and grab out materials and use them. One of the expectations is that everyone shares and everyone takes turn creating new materials. It gives us a lot of materials that we can latch onto later on and accelerate our preparation for presentations. I found that some of our organizational schemes we can share with collaborators. I have a series of collaborators here that do things similar to us, like we do immunostaining or immunoblotting, and we need antibodies. We'll generate a list with key information and ask them to do the same. And then we share that among all of our labs. And if you need to go test a new reagent and know that they have it, it's available to test. This really amplifies what we can do. I hate wasting money on a reagent that I realized wasn't going to fit the purposes that we need. So when a collaborator allows us to just try a little bit, that's super useful. And we enter into the same type of agreement with those collaborators. In fact, we've gone so far as to create a equipment repository, and I've done this across our department. This isn't big scale equipment that we would keep in core facilities, but smaller equipment, just to know that it's available and useful. We have a great collaborative environment within my department and really within the University of Wisconsin-Madison. And so people are very happy to share, but you need to know what's out there. And so we built this list of equipment. We share this with students. What I found that has been very useful, again, if you build a resource, just like we did that with the faculty mentoring document, others might want it. And it turned out that this became very useful for us as we were trying to recruit new faculty to our department, because we could say, you know, we recognize that you're looking for all of these things. We have a bunch of those already. So you could potentially focus your startup money on these other things that would amplify all of our research, and we can work together as a community. And our university has now latched onto that and has created a searchable database across the university for equipment. So really key to doing all of this work, it takes a lot of work to stay organized, to get organized and stay organized. It's writing extra things into spreadsheets, spending more time than you would need to do this if you were only thinking about yourself. It's important that you get buy-in from the members of your team to continue to do this work, because once it falls apart, it's hard to get it back. And so there are many advantages. The more organized we are, the faster they'll be to degree completion if they're a graduate student or an undergraduate. The more publications they'll have, the more they can be successful in terms of getting independent funding. And really, if they're developing good resources, time and again, some of the things that we've developed, others have wanted, and we've shared them, it brings notoriety. And so there's a lot of reasons to buy into this system, but it's important to explain those to your team. Okay, so another part of lab management is building the team and recruiting. I remember when I started, I had big dreams. I'd seen these big, beautiful websites that other labs, big labs, my hero labs had built, and I thought, if only I put time into building a great website, they're all going to come running. Faculty who are admiring the photos and resources and are going to become my friends and collaborators, maybe my Valentines, they're going to come running. Rich people who are going to recognize my brilliance are going to hand me millions in donations. The very best trainees at all stages are going to come and find this lab website and say, I want to work for you. So I put in time, and none of the things that I hoped had happened. And I decided to do Google Analytics to check to see who was actually visiting my website. Against my dreams, not many people. And it turned out that most of them, all of them, were located at UW-Madison. That was super helpful to me because I thought this was my recruiting tool, my tool that would help me build the team that I wanted. And it was to some extent. What I figured out is that the people who were coming to this lab website were undergraduates who were looking for a place to work. And so we scrapped the website that we originally had, and we made a new one. And we made it focused on what we thought undergraduates wanted, needed, would be incentivized by. And I think that that's helpful. So think about what works for recruiting and what doesn't. In terms of other things, building the team, I found that it was much more useful not to spend time on the website for getting graduate student trainees and postdocs, but for actually getting out into the field, presenting work, publishing work, sharing protocols so that others would get familiar with it and say, oh, this is cool if this lab works this way. And so that's where I think your investment makes a lot of sense to take this work and organize it and structure it and make it unified so that you can share it out. And I think that will help you build your team as well. So how do you get individuals ready to go? I didn't want to have to be the one that would train every single person. I like to train people, but I know that I don't have the time day in and day out to do every little thing. And so I put time into building an onboarding document for our lab. And it would be parts of it that I could just highlight that were relevant for undergraduates, parts for postdocs or scientists, or visiting scholars who were just working in the lab for a little while to make sure that they were working safely, working in accordance with the rules that we have here in our lab and at the university, et cetera. And so this onboarding document has been very helpful because I organized it in a way that someone from the lab, not necessarily me, but could be me, can help work through this document when somebody new comes. It discusses logistics. So how do you get access to the building? How do you get access to data? How do our systems work? And how do we have protocols? Where can you find them? How do you save your data? What trainings do you need to do? How do you learn about the safety things like where's the shower or safety shower and eye wash? All of that stuff is listed in there. And it's, again, something else I can share because I think it's worth putting some time into that. Things change. You can update this document as things change, but this has saved a tremendous amount of time by having it be in a way that I know that somebody's going to get good training, even if I'm not the one that's leading that training. I also think it's important to think about how you're going to train individuals in your lab in terms of their learning and knowledge. I remember a very anxious graduate student several years ago came to me and was convinced they were going to fail their preliminary exam, their qualifying exam. And they said, quiz me. Hey, so what do you mean? Well, what are all the questions that my committee members can ask? And so I put together a series of questions. And I said, you should go into the literature. If you don't know these answers already, you should go into the literature and find them. And what I realized was, this is a perfect way to onboard somebody into your science. And we often tell somebody who comes into the lab, go read the literature. And they're eager to do it, but they don't know where to start. And they don't know how far to go out. You can guide their learning just through a series of questions. These are some of the ones I've asked. I've asked different questions depending on which project I think that they're going to be working on. But I say, here's some questions. Go into PubMed, do searches, find some articles, and then write down answers for these questions. And we can talk about them if you have any questions or get excited. Often they get excited about things and new ideas that they can apply to their projects. An additional thing that we're just starting now, and this is really inspired by Dr. Peniston from an experience we had last summer, is to build a guided experimentation resource. And so Dr. Peniston and I co-direct a summer undergraduate research program. And Chris came up with the idea of having a case study that would involve the formation of kidney stones. And she said, having alkaline urine is protective and good for people who are at risk for developing kidney stones. They now sell alkaline water. Come up with a hypothesis and design an experiment to test it. And so the students did that, and we had them work in groups, and they spent quite a bit of time doing this, and they came back to us and said, we're going to do a case control study. Each arm is going to consist of 100 patients. We said, how'd you come up with that number? I don't know, 100 sounded like a good rounded number. And so it provided opportunities to get a little bit deeper into experimental design. Now we train people in the lab. Here's how you pipette. Here's how you pipette for an RT-PCR experiment, but not necessarily the whole process. And so in a course that I'm giving to graduate students right now, we've designed a series of cases for some of the more common types of experiment that they might use in that graduate program. And it's a guided learning assignment. So it gives a scenario. In this case, you completed a bulk RNA sequencing experiment. You found that this one gene was increased by giving progesterone. It was increased in the mammary gland in its RNA abundance, and you want to verify that with RT-PCR. So it goes through the process of, why do you need to do a complementary method? How do you design primers? How do you use a database of genomic information to find sequences and design these primers? And what are the controls that need to go to this experiment? We all have a series of things that we know we're going to do as kind of our bread and butter research. Having this here helps those individuals get trained by going through the process themselves once. And what I think is really important is it forces them to do the research. I'm noticing sometimes if you don't provide resources like this, a lot of times the questions come back to you. How do I do that? Did you Google search it? No, but you could. So this is a good way to get to think about how to do independent research by a guided case study that individuals work on and develop expertise within areas. Okay, I just have a couple of slides left, and then we can take some questions. Last part I want to talk about is budget, and I don't have a ton to say about this, but I remember when I was going to start up a lab, I was very nervous that the startup funding that I was going to be offered wouldn't match my needs. And so I remember thinking, this is what I need to do. I had a K award. I had some aims written down. I went into my current lab and I went through every protocol I needed to do. I wrote down the catalog number of every reagent that goes into achieving that protocol. I went on the Sigma or the Fisher website. I wrote down the cost of each of those things. And then I did that with equipment and I got quotes. And then I did that with personnel to last me for 2 years. And I presented it to my chair in terms of my defense of what I needed for a startup. And he said, you know, it would have been a net fine for you to just say, this is how much I need in reagents and equipment and salary. So I did a little more work than I needed to, but it turned out to be very useful because this was a tool that I could use to populate the lab. So when I first started, I had hired an individual who had a PhD, didn't want to run their own lab. In fact, was interested in going into a different training program, committed maybe a year to my lab until they were able to transition to this new program, but was very competent, had worked as a lab manager previously before going to graduate school, had a shared vision of what we were trying to do in terms of these modified network organizational schemes, and was very trustworthy. So we just took this list of materials and we split it down the middle and we started to do all the ordering. And it was so efficient because it was already there. So took some time, but I highly recommend. It's hard to know exactly what some things will cost. So the way that we track our budgets now is we have an Excel spreadsheet. Every time we order something, we write it down. And we do that for every fiscal year. And so now I can go back over years, most recent years or over the total history of the lab, and know about what we're going to expend for for resources, expendable resources. I can break that down into different categories if I want to. And what's really useful about this is that if you want things to be consistent, sometimes it's hard when somebody's looking something up in an online catalog to get exactly the same thing. Is it 95% peer or 98% peer? You can go back to this list, search for something, and find the exact catalog number of what had been ordered previously, even if it had been used up. So this is a great way to track spending and make things consistent in terms of order. When planning a budget, it's important to realize that you may be getting funding from multiple sources. Maybe they're state sources if you're at a state school like I am, or a state institution. Maybe they're private donor sources, maybe they're federal sources. And there are restrictions in terms of how each of those things can be spent. So it's really important that you understand the types of funding that you have, and the limitations, as well as any expirations. One of the things I came to realize is that certain types of my funding couldn't be used to purchase computers, for example, but others could. It's really good for me to know that. Then I learned that there were intramural funding opportunities, but as long as I had something available in one account, I would not be considered for that. So that told me I should spend that money first. So it helps you to understand the order of operations if you know the restrictions on each of those things. And part of this is actually understanding what your intramural funding opportunities are. They're available on websites, but also from talking to people. So I believe that's it in a nutshell. So the last thing for you to do, enjoy your race. I mean, this is fun. It's fun to get organized, stay organized, and then see your plan come into action.